Hello, I am Christian Buza. In this talk, I would like to highlight key results of my recent paper in which I focused on the classification of gene expression data. First, I will briefly explain the concept of gene expression. Subsequently, I will mention the recently proposed naive Hubness-Bayesian nearest neighbor classifier and the self-training semi-supervised schema. I will highlight that the aforementioned naive Hubness-Bayesian nearest neighbor can be extended to semi-supervised classification, which is one of the contributions of the paper. However, a straightforward extension leads to suboptimal results. Therefore, I proposed a new certainty score in the paper. With gene expression, we mean the process of transcribing a segment of DNA to RNA and translating it into a protein. Roughly speaking, the expression level of a gene means how frequently that gene is transcribed and translated. As we may measure the expression levels of thousands of genes on a continuous scale, gene expression data is high-dimensional vector data. Importantly, a number of diseases and disease subtypes may be associated with characteristic gene expression patterns. Therefore, recognition tasks related to gene expression data may contribute to the diagnosis of various diseases such as colon cancer, lymphoma, lung cancer, and subtypes of breast cancer. Due to the large amount of data, such recognition tasks are typically solved by computers, and state-of-the-art solutions are based on machine learning. Throughout this talk, with classification, we refer to recognition systems based on machine learning. For example, if we show the gene expression levels of some patients with estrogen receptor positive subtype of breast cancer to the computer, and then we show the gene expression levels of patients with estrogen receptor negative subtype of breast cancer to the computer, we want the machine to learn or to recognize the differences between these two groups of patients and be able to detect for new patients if they belong to the first or second group. There are many algorithms for such tasks, such as neural networks or support vector machines. In this paper, I extended a recent classifier, in particular the so-called naive Hubness-Bayesian nearest neighbor classifier, or NHBNN for short. NHBNN performs naive Bayesian inference on the nearest neighbor occurrences, that is, roughly speaking, it describes each instance X using those other instances that have X as one of their nearest neighbors. The presence of hubs, that is, the phenomenon that some instances appear as nearest neighbors of surprisingly many other instances, has been shown to be related to high intrinsic dimensionality of the data. Therefore, Hubness-aware classifiers such as NHBNN are expected to work well for high-dimensional data such as gene expression data. For more information on Hubness-aware classifiers, as well as for an example illustrating how NHBNN works, you are welcome to read our survey or watch the videos on our website. In cases when labeling is expensive and only few labeled instances are present, but we have access to many unlabeled instances, we may want to learn the structure of the data using both labeled and unlabeled instances. When doing this, we say that we perform semi-supervised classification. Self-training, which is illustrated in this slide, is a simple but widespread semi-supervised classification scheme. In this paper, I proposed a new certainty score for self-training that accounts for the presence of hubs, that is, the presence of instances that appear as nearest neighbors of surprisingly many other instances. I performed extensive experiments. For the details, you are welcome to read the paper. Furthermore, please feel free to use our implementation of Hubness-aware classifiers. Thank you for listening.